in our abstract presented here at ICML in Lugano, we show unique abilities of cfDNA profiling in profiling classic Hodgkin lymphoma. Specifically, we show that the mutational representation is actually higher than in the tumor tissue in most Hodgkin cases. And this exquisite plasma representation of this disease we took advantage of and profiled a large cohort of 366 patients using a method called CAPP-seq from plasma samples. And by doing so, we identified 2 genetic subtypes that we correlated with clinical variables, and we found significant associations both with clinical variables and outcome. So specifically, we saw one cluster that had a very high mutational burden and had coding mutations affecting NF-kappa B, STAT6, PI3K signaling, as well as B2M mutations. And another cluster which was more characterized by genomic instability and an abundance of copy number alterations. And we found that the second cluster, the genomic unstable cluster, more frequently affected older patients and more frequently was EBV positive, and also that patients with the H2 cluster had inferior progression-free survival. In addition to that, we proved the biological distinctness of these clusters by transcriptional profiling and found that there were distinct signatures enriched, one a cytokine response signature and a T-cell activation signature. And we could show that the genomically unstable cluster had in fact more T-cells infiltrating the tumor tissue. So overall, we present 2 genetic subtypes that had prognostic correlates and also biological distinctness.